we've seen that the function of DNA is that it tells a cell how to make proteins. That process of making a protein occurs in two steps. There's transcription and then there's translation. We've already taken a look at some of the details as far as what occurs during transcription and what occurs during translation. So what we'll do next is take a look at the other thing that a cell can do with its DNA, and that's to make a copy of it. Remember that the reason why a cell would be interested in copying its DNA is so that when it divides, the two daughter cells that end up being produced has its own DNA molecule inside of it. And so again, we'll take a look at, or we'll review some of the basics of how DNA gets replicated inside of a cell. So if here's the DNA molecule inside a cell that's going to get replicated, the first thing that has to happen is that the two strands have to separate from one another. This exposes the nitrogenous bases, which can then be read in order to make the complementary nucleotide. It turns out that there's an enzyme that is responsible for reading this DNA template and making the complementary DNA molecule from it. That enzyme can only work, though, if it has a starting block molecule that it can work off of. And so that starting block molecule is called a primer, and primers are made of RNA inside of cells. And usually primers are made up, it's a small chunk of RNA, so it's several RNA nucleotides long. But here we're just showing you one of the RNA nucleotides that you find in a primer to simplify things. So after this starting block, after this primer has bound to the complementary sequence on the DNA template, now the enzyme that makes the DNA can build off of it. The name of that enzyme is called DNA polymerase. And remember to keep the different polymerases straight in your head. So when we were looking at transcription, we saw that there is an enzyme called RNA polymerase that's involved. Remember that if it's called RNA polymerase, it's an enzyme that makes RNA. Here now we're talking about making DNA, so you know that the name of the enzyme that's responsible for doing that is DNA polymerase. Again, the term in front of the polymerase tells you exactly what that enzyme is going to make. So after the starting block, after that primer has bound, that will then let DNA polymerase do its job, and basically what the DNA polymerase does is that it helps the complementary nucleotide attach to the each one of the individual nucleotides in the template. So the nucleotide that would bind to A, that would form a hydrogen bond with A, is T. And remember that each one of the nucleotides that are brought in at first have 3 phosphate groups attached to them. And the significance of that is that, remember that phosphates bonded to one another are bonded together via a high energy bond. So whenever that bond is broken, it releases a lot of energy. And that energy is actually required in order to form the covalent bond between this sugar and this phosphate. Right? So the energy represented by those 3 phosphate groups is the energy that's needed to basically hold the sugar-phosphate backbone together. And so that's what this DNA polymerase then does with the rest of these exposed nucleotides. It just helps the complementary one combine and become part of the new DNA that's going to be built, or that's being built. And so at the end here you have a DNA molecule. Again, these sugars and these phosphates are covalently bonded to one another. The nitrogenous bases on the different strands here are hydrogen bonded to one another. The same thing then happens with this strand over here. So it also serves as a template to make a new DNA molecule. In order for that to happen, an RNA primer must bind, and then after it's bound, the DNA polymerase just builds off of that in order to make the complementary DNA strand. The next thing that happens after most of the DNA has been built is that these RNA primers end up being removed and replaced by DNA nucleotides, so that at the end of the process you have 2 molecules, and each of them are made up of nothing but DNA nucleotides. And if you notice, the 2 DNA molecules are identical to one another. So you see the same sequence in this molecule that you see in this molecule. And so basically we've just shown you the general idea of how a cell reads information on a DNA molecule to make another molecule that's identical to it. Remember that bacteria, like all prokaryotic cells, have circular DNA. So in order to copy circular DNA, it looks something like this. The same basic concepts are involved, right? The two strands have to separate. After they're separated, a primer binds, and then that allows DNA polymerase to lay down the complementary sequences. But because the DNA in bacteria is circular, the overall picture of what it looks something like this. So we'll play this for you. It's a video from McGraw-Hill. So actually, I'm not sure if you're able to hear that audio. So what you're taking a look at here is a double-stranded circular piece of DNA that you find in bacteria. The two different strands are color-coded. So you have double-stranded DNA. One of the strands is in blue. The complement is in red. This is saying that the replication of the DNA occurs at the sequence called the origin of replication. So this is where the two strands of DNA are going to become single-stranded so that DNA replication can begin. So this is showing you that when the two strands separate, DNA replication on a circular piece of DNA actually go in two directions at the same time. So what this picture is showing you, well, the dotted lines here represent the new DNA strands that are being built. So as the two new DNAs are being built, kind of the two parts of the circle are being kind of peeled apart from one another. So that at the end you have two DNA strands. They should have completely identical sequences, and again what this is showing you is that the new DNA strands are represented by the dotted lines. So again, the same basic concept. DNA replication involves the strand separating and then a polymerase doing its thing, but the overall big picture looks a little bit different in bacteria just because they have circular versus linear DNA. So what we'll do next to kind of end the discussion on DNA replication is just say a few things about the enzyme that does the replicating, and that's DNA polymerase. The thing that we'll mention about DNA polymerase is that there are actually different types of DNA polymerases that you'll find inside of a bacterial cell. Each one of those DNA polymerases does a different thing, and so we'll spell those out right now. So we mentioned that the major function of DNA polymerase is that it reads a template piece of nucleic acid, and then it helps the complementary DNA nucleotides bind to that template. So the DNA polymerase that does that major lifting as far as the work that's necessary in order to make a new DNA molecule, that's called DNA polymerase 3. So again, that's the enzyme that reads the DNA template in order to make the complementary DNA strand. There are other types of DNA polymerases that are involved in the production of a DNA molecule. So remember that at the end of DNA replication, one of the last things that has to happen is that these RNA primers are removed so that they can be replaced with a DNA nucleotide or a DNA string of nucleotides. It turns out that the enzyme that's responsible for removing these RNA primers and replacing them, that's called DNA polymerase 1, right? So this is a different type, slightly different type of DNA polymerase that plays a role during the making of a DNA molecule. There are other types of polymerases too, and what those other types of polymerases do is that they actually you only see them under certain circumstances. And so there are instances when a cell's DNA might get really badly damaged for one reason or another. And so whenever you have really badly damaged DNA, it turns out that the normal DNA polymerase, in other words DNA polymerase 3, would not be able to read that and construct a new DNA molecule out of it. So if for some reason a cell's DNA becomes really badly damaged, if it's going to try to replicate that DNA, it has to use a different type of DNA polymerase. And so that's where DNA polymerases 2, 4, and 5 come in. So they are special DNA polymerases that are used only when the DNA template for some reason gets really badly damaged. And so we'll actually spend some time in another video taking a look at DNA damage and how that ends up being fixed by these types of polymerases. But just to sum up, what we've just taken a look at here are the basics of how DNA gets replicated inside of a cell.